Good morning, everyone. Myself, Professor Khemchand Surana, Assistant Professor at Divine College of Pharmacy, Satana. So, today we will discuss about the anti-infective agents, that is what the synthetic antibacterial agents. In that, we will see about the quinolones. In last session, we have discussed about the sulfonamides. Today, we will discuss about the quinolones. Now we will move to the introduction. The introductory part is the quinolones are a family of synthetic broad spectrum antibiotic with bactericidal activity. The term quinolone refers to potent synthetic chemotherapeutic antibacterial agent. Quinolones are synthetic antibacterial agents that are highly effective in the treatment of many types of infectious diseases. They are patterned after nalidixic acid, a naphthopyridine derivative, which will introduce for the treatment of urinary tract infection in 1963. The usefulness of the quinolones has been largely confined to the treatment of urinary tract infection. The modern generation of fluoroquinolones, which containing C6 fluorosubstituent, means at 6 position of basic structure there is a fluorosubstituent, and cyclic basic amine moiety at C7 means at 7th position there is a cyclic basic amine moiety, are potent and having broad spectrum of activity. They are used against a variety of gram negative as well as gram positive pathogens. Means they were useful for both gram negative as well as gram positive pathogens. So this is what the basic structure of quinolone. The basic quinolone structure: at first position nitrogen is there and R1 substitution is there. Second position, then third position there is a COOH means acidic group. Fourth position there is a ketone group. Fifth and at sixth position as there is a fluorine group. Seventh position there is a cyclic basic amine moiety with R2 and R3 substituent. Now we will move to the history of these quinolone antibacterial agents. Quinolones were the first developed in the 1960s and can be classified into the generation based on antimicrobial activity. The first nalidixic acid which came in 1962. The first generation of quinolones begins with nalidixic acid in the 1962 for the treatment of urinary tract infection in humans. Nalidixic acid was discovered by the George Lesser and the co-workers in the distillate during an attempt at the chloroquine synthesis. A group of synthetic antibacterial agents mainly effective against gram negative bacteria. Nalidixic acid first member of introduced in the 1964 for the urinary as well as GIT infections. In congeners oxalinic acid and rosoxacin with more potency in 1970s. The second generation, which will also called fluoroquinolones, with extended spectrum and systematic effects in 1980s. Since then, many synthesized with useful spectrum. Now, this is what the classification of quinolone antibacterials. which will base on the generations. There are four generation: first, second, third, and fourth, having an examples with antibacterial spectrum. So the first generation nalidixic acid and cyanoxacin example having a gram negative bacteria aerobic antibacterial spectrum. The second generation includes norfloxacin, ciprofloxacin, ofloxacin, lomefloxacin, inoxacin, having an antibacterial spectrum, gram positive bacteria, aerobic, and improve activity against gram negative bacteria. Third generation levofloxacin, sparfloxacin, glatifloxacin, gemifloxacin, which will having a antibacterial spectrum with good activity against anaerobic gram positive bacteria, particularly pneumococci. Now come to the fourth generation examples: trovafloxacin, moxifloxacin, clinafloxacin, sitafloxacin, anaerobic, increase activity against pneumococci. So this is what the basic classification. You need to remember the generation with their examples. Now move to the chemistry part. That is what you need to remember the structures, as well as how to remember, means the practice you have to need. The first generation having example nalidixic acid and cyanoxacin. You have to write these structures and practice them. Now second generation inoxacin, ciprofloxacin, ofloxacin, norfloxacin. The ciprofloxacin synthesis has been asked in the final examination. So you have to remember the structure as well as their synthesis. Also the ofloxacin, which is what the racemic mixture, and leofloxacin in pure S isomer form is used as an antibacterial agent. Now the third generation sparfloxacin, you have to again write the structures and practice. The fourth generation moxifloxacin and gatifloxacin. So the students need to write all the structures and draw all the structures with practicing. Now we will move to the structure activity relationship of quinolones. So in the center there is a two structures. The first structures having the acetic group with quinolone, and at fourth position there will be a oxo group, means ketonic group, and at nitrogen there is a R substitution. And second structure which is having this basic structure of first and substitution at every carbon. The first point is the study have shown that the 1,4 dihydro 4-oxo 3-pyridine carboxylic acid moiety is essential for antibacterial activity. So whatever this basic moiety which will given in the center of slide, which is essential for the antibacterial activity. The pyridone system must be annulated with an aromatic ring. The pyridone system means for the pyridine having a ketonic group which will be annulated. Annulated means for the merge, which will fuse with what aromatic ring. So this is also required for activity. The second point is the isoteric replacement of nitrogen for carbon atoms at position 2, means what the Quinolone. If we will isoteric replace the nitrogen for carbon atom at position 2, the structure will came as a Quinolone. 1,5 naphthopyridine. If we will replace the fifth carbon with nitrogen, it will the 1,5 naphthopyridine. Now if we'll replace at sixth position, it will 1,6 naphthopyridine, and if we will replace at eighth position, which will the 1,8 naphthopyridines, leads to the retention of antibacterial activity. Means for the if we isoterically replace these carbons with nitrogen at respective position, which will leads to retention of antibacterial activity. Now here the initially examples are given with basic structure and the example of sparfloxacin. Now the third point is the introduction of substituent at position 2 greatly reduces or abolishes the activity. So already you know that first position there is R1 and subsequent there is a R2. If we will substitute the any type of substituent at R2, it will abolishes or reduces the activity. But the substitution at position 5, 6 and 7, you can see in the basic structure there is substitution R5, R6 and R7, and 8 of the annulated ring means the substitution position at annulated ring of the annulated ring means the aromatic ring, which will fuse with your pyridine, produces the good effect. Means what? If substitution at these position 5, 6, 7 and 8, it will produces the good antibacterial activity. Here the some examples is given. For example, the piperazanil and 3-amino pyrrolidinil substitution at position 7 enhances the activity against Pseudomonas aeruginosa. If we will substitute these piperazine basic moiety or piperidine basic moiety at position 7, it will leads to a increase the activity. Amino group at C5 position increases the activity. If at fifth position we will introduce the amino group, you can see into the example of sparfloxacin at fifth position there is an amino group which will increase the activity. For example, the sparfloxacin, which is given into the tops left slide of your left part of your slide. Now next point, fluorine atom substitution at position 6 is also associated with significantly enhanced antibacterial activity. If we substitute at position 6, fluorine is also enhance the antibacterial activity, means it will helpful. So on the basis of these, the next generation, that is we can call the fluoroquinolones, were developed. Now the fifth point, the alkyl substitution at position 1 is essential for activity. At R1, which is substituted on nitrogen, this may be the alkyl substitution which will essential for the activity. Lower alkyls, for example methyl ethyl cyclopropyl compounds, generally having greater potency. So we are having examples, for example cyclopropyl in ciprofloxacin having a greater potency as well as activity. Now aryl substitution at position with also consistent with antibacterial activity. If we substitute any kind of aryl ring on this position, for example ofloxacin, it will having a consistent activity. Ring condensation at position 1 and 8, 5, 6, 6, 7 and 7, 8 position also leads to active compound. So condensation is also helpful in the case of fluoroquinolone derivatives. Now this is what the summary of this SAR is given over here. Now you need to remember these points. So again, in the center there is a basic structure is given. At R1 initially we will see substitution, we will see through the substitution pattern. So at R1 there is a square box is given. In that the written as the increase overall potency. So how it increase the overall potency? So there is arrow, and above there again there is one box and having these aryl substituent. Already we have seen the same point into the SAR. Again, if we will substitute any kind of alkyl substituent at R1, it will increase the overall potency. Here the example is given. Now at C2 there is nothing. C3 there is COOH, C4 ketonic part. Now come to this R5. In R5 the square box is given increase gram positive activity. Above that there is a NH2OH and CH3 sequence is here. If we substitute these kinds of groups over R5 group, it will increase the gram positive activity. Now R6 increase gram negative activity when there is a fluorine group is substituted at R6. Now at R7 increase activity of gram negative as well as gram positive. Now at R7 position both positive as well as negative when these heterocyclic rings or aromatic rings were substituted on the R7, it will increase both positive as well as negative activity. Now the last is what the 8 R8 increase the activity of anaerobic and gram positive bacteria. So when these groups which will given into the blue color as well as red color, you can see into the box the anaerobics is given into the violet color and the substitution is also given into the violet color box. Gram positive also the blue color and the substituents and also into the blue color boxes. So this is what the summary of structure activity relationship. Now again the similar thing with different points. At R1 increase the volume of distribution and viability. So these are the some points where also important as per the structure activity relationship. At R1 increase volume of distribution and viability. So increase viability, half life and decrease resistance in case of 8 position. Now come to the 5th position, increase the lipophilicity when it will substitute with NH2. At R6 increase the volume of distribution and tissue penetration when there is a fluorine is substituted at R6. Now R7 increase half-life and CNS penetration when these heterocyclic compounds were substituted in this sequence. Also the substituent which are substituted on basic structure which will having a corresponding mechanism for their activity. So we can discuss about the same. If the substitution at R1 which control the potency and some effects on pharmacokinetics. If we will anything substitute on R1, it will controls the potency and some effects on the pharmacokinetics. R2 close to gyrase binding site means when this R2 site is very close toward the gyrase binding site. As in next slides we have to see about the mechanism of action. This mechanism of action is basically based on the inhibition of DNA gyrase enzyme. So this site basically R2 is close to the binding site. Now this acidic as well as ketonic part essential for the gyrase binding and bacterial transport. Now R5 control potency adds gram positive activity. R6 means fluorine substitution controls gyrase and bacterial potency. R7 controls potency, spectrum, and pharmacokinetics. 8th position controls pharmacokinetic and anaerobic activity. So these are what the three different summaries we have seen. Initially, the first is what about the structure activity relationship, second about their pharmacokinetic and dynamic related, and the third one is about the mechanism of action. So now we will move to the mechanism of action of quinolone antibiotics. So the antibacterial action of quinolones results from the inhibition of DNA synthesis. So major focus mechanism of action of quinolone antibiotics, or you can call antibacterials, is the inhibition of DNA synthesis. So how it will inhibit the DNA synthesis, we have to see. So this effect is caused by inhibition of bacterial DNA gyrase. So how? So DNA gyrase is enzyme which is helpful for synthesis of DNA. We have to inhibit, means whatever the medication quinolones have to inhibit the bacterial DNA gyrase, which will also called as topoisomerase second, an enzyme responsible for introducing negative supercoils into circular duplex DNA. Means what? This enzyme is useful or responsible for the synthesis of DNA. The fluoroquinolones inhibit the enzyme bacterial DNA gyrase. So these fluoroquinolones inhibit the bacterial DNA gyrase, primarily active in the gram-negative bacteria. So these DNA gyrase primarily active in the gram-negative bacteria. This point should be asked in GPAT examination. Which nicks double-stranded DNA, introduces negative supercoil, and then reseals the nick ends. This is the necessary to prevent excessive positive supercoiling of the strand when they separate to permit replication or transcription process. So what these agent has to prevent the excessive positive supercoiling. This supercoiling when happen when there will be the DNA synthesis. So this supercoiling has to be prevented by these quinolones and separate to permit replication and transcription processes. The DNA gyrase consists of a 2A and 2B subunit. These DNA gyrase enzyme having a 2A and 2B subunit. The A subunit carry out nicking of DNA and B subunit introduces negative supercoiling, and then A subunit reseals the strand. The both A subunit as well as B subunit having a particular role. So A subunit carries the nicking of DNA. So for nicking of DNA, the position at A subunit, and B subunit introduces negative supercoil, and then again the A subunit reseals the strand. Fluoroquinolones binds to A subunit with high affinity and interferes with its strand cutting resealing function. In gram-positive bacteria, the major target of fluoroquinolones or quinolones action is similar enzyme, means topoisomerase 4, which nikes and separate the daughter DNA strand after DNA replication. Greater affinity for topoisomerase 4 may confer higher potency against gram-positive bacteria. The bactericidal action probably results from digestion of DNA by exonuclease, whose production is signaled by damaged DNA. In place of DNA gyrase or topoisomerase 4, the mammalian cells possesses enzyme topoisomerase second that also removes positive supercoils, which has a very low affinity for fluoroquinolones or quinolones, hence low toxicity to the host. So now you can see the structural mechanism of action of quinolones. Here into the 3D structure, the ciprofloxacin structure is given, means for the quinolone antibacterial, and below there is a DNA synthesis process. So initially what the bacterial cell is there, in that the supercoiled DNA. So this bacterial cell consists of supercoiled DNA, which will converted into or synthesize the bacterial chromosome through the DNA gyrase enzyme. Further, this bacterial chromosome will replicated through the DNA replication process, and further by topoisomerase 4 there is separation of daughter chromosomes. This is what the basic process of DNA synthesis. Now this quinolone, you can see the negative sign or minus sign over here. So these quinolones will inhibit these DNA gyrase as well as topoisomerase enzyme. So now come to the adverse effect, or you can call the side effects. So this table has been given as per for your GPAD examination. So you have to or need to remember all these side effects of particular quinolone antibiotics. So just we will see the gastrointestinal effect will produces by sparfloxacin, gripafloxacin and others. Arthralgia effect, sparfloxacin, leofloxacin, gripafloxacin and others. CNS effect, trofloxacin, leofloxacin. The phototoxicity, you have to remember the phototoxicity questions may ask in GPAT examination. The phototoxicity will caused by the clinafloxacin, sparfloxacin and others. Further, the genotoxicity also important, lomifloxacin, moxifloxacin, ciprofloxacin. Then QTC prolongation. So initially whenever you are studying these side effects, you have to know the meaning of these all side effects, then you will remember these side effects with examples. Then further hematological effect, timofloxacin, hepatic eosinophilia effects, trofloxacin, gripafloxacin, others. Pulmonary intestinal, gemifloxacin, immunological side effect, gemifloxacin, hyperglycemia, cinefloxacin, gatifloxacin, and CYP 440 450 inhibition. So these examples. So you have to remember this as per your GPAD examination. So further similar thing is given over here. So you have to remember these adverse effects as per the CNS central nervous system. The quinolones may displace the neurotransmitter GABA. So these quinolones will displaces the neurotransmitters GABA, resulting in the CNS stimulation. They will causes the CNS stimulation by replacing the GABA. So dizziness, headache, insomnia, restlessness. Then further phototoxicity, skin damage after exposure to UV light during toxic reaction. If you have consumed the quinolones and you were exposed to UV light for more time, the skin damage may occur. So these include second degree burns, discoloration, sometimes permanent discoloration and scarring of skin. Then cardiac toxicity. So cardiac toxicity, this is what the important as per your GPAT examination. So the torsade D point, this is what the side effect. Paroxysm of ventricular tachycardia, in which the electrocardiogram shows a steady undulation in the QRS axis in runs of 5 to 50 beats with progressive changes in direction. It is the most severe type of arrhythmia and can result in death. It is the most often associated with and preceded by prolongation of QT interval. So this is what the torsade D point you have to remember. Now GI related, depletion of magnesium, disruption of cellular enzyme function, disruption of mitochondrial function and energy production. Other includes nausea, vomiting, anorexia, diarrhea. These are commons. So now in final examination, there will be a question asked about the synthesis of ciprofloxacin. So you have to remember, you have to practice, note it down many times and practice it. This synthesis of ciprofloxacin. In final examination, there is a question for 10 marks, which is like that: the explain the classification SAR, synthesis of ciprofloxacin, and adverse effect of quinolone antibiotic, or explain the classification SAR ADR of quinolone antibiotic with synthesis of ciprofloxacin. Or for in a short question, maybe ask for 3 marks or 5 marks, synthesis of ciprofloxacin. So here the synthesis of ciprofloxacin is given: 2,4,5 trifluorobenzoic acid is what the starting material. The first intermediate were synthesized by these two starting materials, which is COCl, COCl2 and DMF solvent. Further the O2 means what? Your lithium containing particular catalyst has been used. The first intermediate we will ask further in transition state. So cyclopropylamine with tertiary butoxide, get these intermediate further with potassium tertiary butoxide and potassium tertiary hydroxide use. The cyclization will occur with HCl and acetic acid means ACOH. You will get these basic nucleus. Further with the cyclic amine treated with this intermediate, you will get the ciprofloxacin. So you have to write this and you need to practice. So here we have completed this quinolone antibiotic. So you have to now make the notes of whole chapter. Whatever the structures are given, synthetic part is given, structural part is given, you have to write it, you have to practice it. If any kind of query, you can ask me in my contact number. Thank you. Wish you all the best. Happy learning.